I was being asked to give a brief workshop about network biology, and I was having in mind mostly the community that essentially have rather less quantitative background, which I think is not necessarily the case here. It's actually heavily charged into the quantitative part, but maybe not necessarily using graph theory. So having that in mind, if I am going into not too many mathematical details, it's just out of the sake of focusing on the biological aspect. So in terms of the basic concepts, before defining actually what a network is, which actually I think a vast majority of people having one way or another seen networks, starting, for example, by social networks. I wanted to start just by the question of why do we need to think in terms of networks when we want to study biology? And I think that one short answer is from the point of view that biological systems are complex systems. So complex systems do not mean complicated. It means complex in the sense that they tend to be of large quantities. For example, if we are trying to study gene regulatory networks, but then also the fact that there are interactions among them, and because of those interactions, then it is going to be hard, typically, if we want to know, for example, if we perturb a cell, or in this case, we perturb a gene regulatory network or a gene regulatory system, just by knowing information about the independent parts of the system is not going to be enough in order for us to understand the behavior of the system as a whole. Furthermore, another thing that defines a complex system, which is the case of biological systems, is that because of those interactions, then some properties are going to emerge. The other thing that also occurs typically with biological systems is the fact that if we do a perturbation in one part of your system, does not mean that the whole system is going to collapse. There is going to be some kind of robustness. So all of these characteristics are typically what are defined into a complex system. And as such, you can imagine that by having information as super useful as can be, to have information about every single element of your system, to just have it as a list of information or as measurements is not going to be enough to draw conclusions. More importantly, even, for example, to understand overall behaviors and or even to manipulate. And so it is important to rather understand the system as a whole, so not only the components, but actually how they interact with each other. Typically, when I talk to students in their first years or that are in the field of genomics, for example, I just make a caution note on the perspective that while, for example, in bioinformatics, it's super useful to take measurements. For example, you do DNA sequencing, you do gene expression, and you might understand, what a gene does under different conditions. It goes up, it goes down in terms of expression. That is wonderful. That's called genomics. You can study this at a genome-wide level. That doesn't make it system genomics. It makes it system genomics the moment that not only you are talking about the different components, but how they interact with each other. Furthermore, which goes a little bit further than network biology, how this happens actually throughout time. So now we're talking about dynamics, but we're not going to focus on that today. So that is why we need to think about networks when we want to study complex systems. So what is a network? Well, a network is simply an object that has two types of objects. One are the nodes, like in this case, these 6 nodes, 1, 2, 3, 4, 5, 6. So these are typically biological components, so genes, proteins, metabolites in the case of intracellular networks. And the second type of objects that define a network are links. So these are interaction among the different nodes, which typically, in the case of intracellular networks, they might be representing regulation, physical binding, or reaction. Now, networks, for mathematicians, as you know, are rather known as graphs right? And so this idea of graphs and this field of mathematics has existed since the 1700s at least, right? One typical example that we have was precisely with Euler, who was trying to solve this problem of the Königsberg bridges. So try to identify ways to move across the city by crossing the bridges only one time. And one way that he realized that he could solve the problem, which by the way, it turned out that it has no solution, to pass only one time to go to the different points of the city, was to represent this city map by representing the different regions with vertices, and the fact of crossing from one bridge to another to represent them with edges. And so this was one of the first moments that we have documented of the emergence essentially of this new field of graph theory. And there are so many tools and so many metrics that we can use in graph theory that can totally be transported into the study of biological systems once we have represented them as graphs. So going back, so a graph, as we have said, we have an object. So a graph G has these two sets of elements of vertices, which we call them before as networks nodes, and a set E of edges, which also we are calling these links. So what kind of things can we look at just as a first super basic level? So we can have networks of two types. We can have them directed or undirected. So in the case of directed, the edges are going to have a direction, for example, from A to C, as you can see in this red arrow. And we rather, in that case, when we are showing directionality, we are trying to portray some kind of cause and effect, some kind of causality. And so this typically is represented, for example, in gene regulatory networks or in signaling networks where we actually care about the direction. But then also we can have undirected networks on which edges are rather associations or correlations, for example. And so in that case, we represent symmetric relationships. Typical example of this is when we see protein-protein interaction networks, two proteins interact with each other, there's no particular direction. So we just paint them there as undirected networks. Also, networks can have now different types of features assigned into either their edges or into their nodes. Here is one example on which we are assigning features, in this case, number values, into the edges. So we can have them unweighted. So the links simply show that a relationship exists. But we can also add some kind of weights. For example, let's say that we are trying to infer a gene regulatory network and we are trying to show some kind of confidence that such interaction exists between two genes. Or maybe we want to talk about signaling networks and we want to show some kind of a strength of a signal. So in that case, we will actually add numbers into the edges. Other important objects and metrics that we can use are paths, cycles, and connectivity into the networks. So paths are a sequence of vertices where each vertex is adjacent to the next one. And so then we can add some kind of metrics into those paths, right? So we can say shortest paths, the longest path, and then we can define characteristics on the graph that we might be studying, for example, the diameter, because now we are measuring all of the different lengths of paths between different nodes in the network. A simple path is a path that does not have repeated vertices. A circuit is a path that begins and ends at the same vertex. And a cycle is a circuit that doesn't repeat vertices except for the start and end point. So in the case of graphs that are representing, for example, in the case of discrete systems, when we are trying to represent dynamics and how the system is evolving from one state to another state, in this case, these cycles are very important because are showing some kind of stability or some kind of cyclic behavior. So extremely important and useful representations. Now, in terms of connectivity concepts, a connected graph is essentially a graph where you can get from any node to any other node by means of passing through the different vertices that you might have. A component is a disconnected graph that can be split into a number of connected components, and strongly connected component, it is in the case of directed graphs, is where you can actually paint a direct path from one vertex to any other one. And again, these components, even in the case when you are studying dynamical systems, you can have representations on which you know that you have basins of attraction. And so to know whether, a particular state of the system belongs to one component or another gives you so much information about ultimately what is going to characterize your cell phenotype. And so in terms of biological networks, we can have different types of scales that are going to be used essentially to build any of these networks. On the left-hand side, I put examples of intracellular networks, that is, networks that are representing objects that live inside of the cell. Examples of those would be gene regulatory networks, signaling networks, metabolic networks, protein-protein interaction networks. So in the case, for instance, of gene regulatory networks, you can have two types, one on which the nodes are simply genes, and the interactions between them are indicating activation or inhibition between them. So in that case, you will have them directed, and you will have them with a sign, so positive or negative, whether it's activation or inhibition, respectively. In the case of signaling networks, you can rather have not only genes, but you can also have proteins and complexes associated into these networks. And in the case of metabolic networks, you can have also metabolites. In protein-protein interaction networks, you obviously have proteins and interactions between them. But then you can actually go out of the cell and have other kinds of scales. So, for example, you can have drug-target interaction networks. You can have disease networks, neural networks, not in the sense of AI, but in the sense of brain networks, right? Cell-cell communication networks, and many other ones. Sometimes they do not necessarily have to be representing, when you go at larger scales, it doesn't have to be necessarily representing anatomy. So one example of this would be functional imaging interaction networks. So essentially measurements on the brain when you do an fMRI scan, and you are seeing what different regions of the brain are on and off. And so the interactions, the way that you break out that those networks are just by voxels, essentially volume pixels. You break down the whole image, and then you start drawing interactions between the different brain regions that get on and off according to a particular stimulus. So on these different scales, you can, now that you have them represented as graphs, you can study different kinds of things on them. But before going into that topic, I will touch on the first natural one. Okay, so it seems that if I have the interest of studying a particular biological system, being a complex system, it seems that is a good idea to represent it as a network, first to start understanding it, to then study it, and then maybe even learn some ways to manipulate it and get something out of it. But how do I reconstruct it in the first place? In a nutshell, we can go to do that into two different ways. And I have to say, nowadays, we rarely do either of the two extremes if we want to build larger scale networks. But just to kind of make you understand these kind of two approaches. So one is what is the bottom-up approach, and the other one is a top-down approach. In the bottom-up approach is actually a lot of what we saw in the previous talk, which is you have information about your system, you make some assumptions, and then based on that, you start building your network. So in this case, you have your different species, and then you're trying to understand, for example, in the case of a food web, what are the interactions between them, who eats whom, how many of those elements you have, and then you do the model of that. So you are doing assumptions ahead of time. You might have information of that, and then you build your network. Once you build a network, and you start studying, and then maybe you realize that it doesn't quite fit the reality, so you adjust it based on that information, and you go back, you revise it, and so on and so forth. So that would be a bottom-up approach. In the case of the top-down approach, it's now on the other extreme, which is we assume no information whatsoever ahead of time. The only thing that we have are measurements. Obviously, from the moment that you are doing measurements, you are doing some assumptions, right? Because you are deciding what are you going to measure, and you assume that those measurements are going to give you all the needed information for you to study the system in the first place. But, okay, you take measurements. So, for example, gene expression data you might have, or perturbation data. And taking that data, you use it in different ways in order to construct your network. Then you go and check it, test the network, and then based on that, you might want to correct it and so on. Actually, yesterday was one of these topics, right? The inverse problem is exactly that. It's how to reconstruct your network, maybe even your dynamical system based on data. So here is one typical cycle for this top-down approach. So you have your data and your measurements, and so you can actually infer two types of objects. One is the static graph, what we just talked about, which is in the case of gene regulatory networks, your nodes are going to be genes, and based on the data, you are trying to infer essentially which genes interact with which other. But this is essentially a static graph, so a directed static graph, maybe weighted if you want to assign some kind of certainty into the interactions that you have. The second object that you might want to infer when you have data is not only the static graph, but actually the actual dynamical system. So this is an example here of an ODE where now you are going to use the data in order to try to infer some of these parameters. And there are computational methods that actually can do either only one and or both, so the static graph and the dynamical system. And once you have that, you can do simulations, and then based on those simulations, see if they match with the data that you might have had, maybe data that you might have hidden, or additional experiments that you might generate. You might identify some errors, and so then you might want to fix it, and then you keep going on to that reiteration, essentially, or iterative process. And as I said, there are many approaches to do so, and as we essentially work with network reconstruction, the reality is that we rarely do these two extremes, right? We tend to do a combination of both. So sure, we have a lot of data, if possible, but then we acknowledge the information that we have learned in biology. So maybe we consult also databases, utilize other sources of data that might be useful, and combine the two in order to help us to do those inferences. And now with AI, many more methods are actually coming up to do even those combinations even better. So here is a set of different methodologies, to which actually I want to bring your attention that for several years, many methodologies were being proposed, computational methodologies to do this inverse problem. And up to 2004, 2005, the community arrived to the realization that in reality, there was not a good benchmark to say, you want to propose a new method for the inverse problem of inferring gene regulatory networks. What would be a good benchmark to say that your method is better than any of the ones that existed before? There was not such a thing, and so the community got together, and I think 2006 was the first competition organized by what's called the DREAM competition, so the Dialogue for Reverse Engineering and Assessment Methods. And throughout several years, I think up to 2012 or 2013, they continue doing several competitions on that. I recommend you a lot this paper, Wisdom of Crowds, even though it was done in 2012. I think that this was an extremely important lesson that I continue seeing the failure of us as scientists recognizing that learning, both for the inverse problem and for many other problems of people like myself who are creating algorithms for analyzing biological system. And the lesson is we can always have amazing ideas of how to solve one of these mathematical problems to study biological systems. In the case of the inverse problem, what we learn is that most likely we will never have the method that is going to be the best one for every single type of data. Even if you have gene expression data, the different systems might have different types of challenges, hidden variables, some kind of coupling information, so on and so forth. So even with the same type of data set, so gene expression, for example, you might have different methods that work better for different characteristics inherent in your data set. Therefore, one very smart thing to do is to consider the combination, so the crowdsourcing of answers through different computational methods. And that was one extremely important lesson, I think, that we learned and that I still don't tend to see still today in many methodologies to apply that knowledge. And but okay, there are many methodologies, and as I mentioned, nowadays it is extremely important now with so many databases that are created at larger scales, and with AI, now this is becoming also very interesting, and also with data that are now at the single cell level, even more challenging, but also more interesting. Okay, so let's assume that somehow, whether it is by utilizing our preferred computational tool or by combining several of them until we are happy with the network that we have built, what can we do with it besides already understanding characteristics? And so I have to say, one of the things that we can do when we have networks, already correlation studies are extremely useful. So I did not write it into my PowerPoint presentation, but just to kind of allude to the fact that causal interaction in networks, of course, allow us to make a lot of inferences, but also already correlation interactions and networks that are drawn on correlation interactions can already give us a lot of information. One example of this is in neurobiology, essentially, in which, for example, neuroscientists have compared these brain interaction networks and compare them across what we will call non-patient data versus patient data, for example, with ADHD, with dementia, with Parkinson. And so some differences in the structure of these brain networks have been observed. So while it's not perfect and while it's drawn through correlation, I think that is definitely, if not much better, at least excellently complementary to a lot of the qualitative data, the clinical data that essentially neuroscientists would tend to utilize in order, for example, to even diagnose patients. And so in this case, there are many publications on which they have realized that there are very clear differences in terms of the topology of these static networks. In this case, I decided to go through one example of how can we use networks for one problem that is dear to mind, which is cell reprogramming. So what do we mean by that? Going back into controlling systems. So what it means to control a system, here I am drawing essentially this metaphorical representation, right, of the Wannington landscape, in which we imagine that we have a cell. In this case, in the original version in the 1960s would have been a cell that is in a stem cell-like state. And this metaphorical representation was used to say that as the cell starts differentiating, it will eventually arrive to its ultimately differentiated state, which then will be represented by these different values where they are essentially stuck and they hardly can get out of that. We know nowadays that this perspective is no longer realistic in the sense that now we have actually experimental tools. One example would be the 4 Yamanaka transcription factors that we can apply into somatic cells that actually allows them to reverse back into a more stem-like state. That is what is called induced pluripotent stem cells. So we actually can go backward, and this is not necessarily really a mountain with one hill, but it's just a mountain with many different hills. And this is what mathematically we can represent as an energy landscape, which is not a trivial thing to do. Anyway, so when we are thinking now in terms of systems biology, this metaphor actually has been translated into several explicit mathematical objects. So what that means essentially is that in this case, a cell, for example, that I can represent, let's suppose that we are talking about a gene regulatory network. I have my system and I see how it changes throughout time. For example, when I do a perturbation. And so eventually the system tends to arrive to some kind of either cyclic or stable steady state or an attractor. And the great advantage of that, at least in the terms of the stable attractors, is that biologically speaking, we can associate them to observables. So to a cell phenotype that we can measure. And so what it means to control, for instance, this system, what it means is that I might have my system in a particular state, an initial state, 0, that I know that in its longer term is going to arrive to an undesired attractor state. So to control the system would mean that I can identify perturbations, in this particular case for cell fate control, it would mean that I can perturb my system at that initial state so that I send my system into a new initial state, X prime, which longer term is going to be associated now with a desired attractor and therefore to a desired outcome. So this is the problem that I am very interested in my lab, to identify methodologies and to do applications for different applications. And so the second word of caution that I think I've been alluding into the previous slide is the fact that while networks give us a tremendous amount of information, it already helps us to understand a lot about our complex systems, is not enough information. The dynamics is also extremely important. In other words, what changes happen throughout time. And the same for how are we going to control these systems. And so what it means is that when we are drawing a network, we are drawing essentially just a snapshot of the system at a given time. But if we want to understand how the system evolves throughout time, how it responds to a particular perturbation and eventually reaches back some kind of stability, we definitely need to study furthermore the dynamics. And so this is one example of one of those computational tools that we created. Yes?
Time, can you wrap up in the next?
Oh, yes. So in this case, we actually propose a methodology called NITMB, in which we utilize essentially gene expression data on which we will have measurements of the desired and undesired phenotypes, and then utilize what is called the feedback vertex set control in order to identify these set reprogramming points. We apply this method essentially to several examples that have been experimentally validated, and we were able to identify the same targets. And so what I more or less have alluded here into this talk is the fact that altogether, network biology can help you to shift essentially the focus from individual components to interconnected systems, and that we can use a lot of the concepts of graph theory to model complex biological systems, which then in turn is going to allow us to do an analysis of system-wide changes, for example, in health and disease, which is obviously going to lead us to a deeper understanding of targeted interventions for our interest. All right, so I think that this is it for me.
Time for a few questions.
Yes. So for the benchmarking, right?
Yeah.
I would imagine different methods probably have trade-offs. For this, so are there like a battery of tests to see whether you're better this for the worse than in the other case?
There was, so what it was being done was not to measure onto a decision of why one method was better for one particular data set. What they actually would do was to categorize the different challenges. Essentially, they generated in silico data, or actually gave data from different biological systems, and then they put the data out there, and then teams around the world would just take this data, and then we will essentially infer the network, giving them essentially the network with the nodes, and then with weights on the interactions according to some kind of certainty that we will have. And then the organizers would say, this obviously would be because they already have the computer that created the in silico data, or because they have worked with these biological systems, and then just tell us, okay, you did well or not so well based on precision recurrent curves and area under precision recurrent curves. So that was the way that they were categorizing better or worse methods according to these different categories. But other than that, there was not a particular map, essentially, of, these methods are better if you think that you have hidden variables. No, unfortunately, nothing of that is measured. If you are curious about that, if you go to the DREAM website, you can download yourself all of the data sets, the different methods, and in the papers, you can see a lot of the results. If you are curious, actually, to study more in depth of what characterizes better and worse answers, or why they got better or worse answers in different types of data sets.
Thank you.